We finished last time on digestion, and I'm going to take a jump to plate 113 for lesson 19. And if we have time, we'll come back and discuss aspects of the neural system. But I'd like to focus now on endocrines. The two systems, neural and endocrine, act in concert. The neural system is a fast, responsive system. The endocrine system, much slower acting, but prolonged. Let's kind of take a general overview. We'll walk through and talk about endocrines in general, and then we'll come back to deal with specific endocrine organs, glands, the endocrines that are produced, their target tissue, and their actions. So on plate 13, if you look under the endocrine glands, you can think, well, it's if you put long ears on this, it becomes a dog, So, but when we talk about glands which produce chemical moieties that are released into the blood, we think about the pineal gland, which is located in the brain, as is the pituitary, and as we will see, the hypothalamus is also involved. And over here, these are organs with part endocrine functions. The hypothalamus we've talked about in terms of being a homeostatic regulator. It also plays an important role in endocrine production. Further down, thyroid gland. The thymus in the thoracic cavity has some important roles, particularly in young animals. The heart, we've in fact talked about atrial natriuretic peptide. That is an endocrine that is produced by the heart. So in addition to being a cardiovascular pump, it is an endocrine organ. Oh, I forgot, we have to go back up to the neck. Before we get into the thorax, we have the thyroid gland in the neck, as well as the parathyroids. Move down, the liver has function. We've talked about hormones of digestion, so you know that they're hormones produced by the stomach, hormones produced by the pancreas. In addition, there are glands that sit just above the kidneys. These are the adrenal glands. We've actually talked about that, aldosterone. So you've heard some of these. And then the ovary and the testes. We will probably save the discussion of endocrines in the reproductive organs for the specific topic of reproduction later. And if we look over on the right side of plate 13, these are organs that have endocrine function invested in them. Hypothalamus, liver, thymus, heart, kidney, stomach, and then the duodenum, right? Cholecystokinin. Well, you have these cells that are producing these hormones, and the original concept was that this hormone was dumped into the blood. It circulated throughout the entire body and then had target organs. So that's kind of the classic definition of endocrine, and that's represented in the first represented cartoon. You have the cell which is elaborating or producing this endocrine moiety. It gets dumped into the capillary system, carried by the veins to the heart, and then is distributed to a target cell. That is the cell that is going to perceive the presence of this endocrine, meaning that this cell has to have some type of specific receptor. And we'll talk about those as well. We'll see that many of the hormones act in series. So a hormone may be produced by one tissue. It will activate hormonal release in another tissue. So the target gland for this hormone is this tissue, and then this tissue elaborates yet another hormone which is released into the blood, again circulating throughout. Neuroendocrines are hormones which are produced by neural tissue. We've talked about one already, epinephrine. We used epinephrine as our sympathomimetic, that is, it mimicked the functions of the neurotransmitter norepinephrine, but it was produced by modified cells and secreted into the blood. So the adrenal medulla is producing epinephrine. So that's a neural cell that then dumps its hormone into the general circulation, and it produces some effect on its target tissue. Here's an example in which there is a portal vessel. That is, the neurohormone is released into a portal vessel. So there are capillaries here. The hormone is released. It then goes and into another capillary bed and released. Paracrines are local tissue hormones. And one of the best examples of this are the prostaglandins, cells which produce a prostaglandin, an inflammatory-like substance that has very localized effects. So they may not be using the blood for transport, but may be moving into the interstitium and affecting. We also recognize that some hormones feed back on the very cell that they are producing. And that would be an autocrine. So paracrines, very localized activity. Autocrines, feeding back on themselves. Let's now turn to plate 114 and look at how hormones act. We're going to divide our hormone population in two. We're going to talk about hormones which are protein derivatives and hormones which are lipid or steroid derivatives, because the mechanisms by which they act, that is not plate 114, is it? Yeah, this is what happens when you let your professor scan the documents. There we go. So on the top of 14, we talk with a hormone cell. We talk about a hormone cell which releases its hormone into the general circulation. Now, many of these hormones are not transported free. That is, they have a carrier. And in this specific example, there are proteins that are involved in picking up this hormone and helping transport it. In part, to reduce the degradation of this compound. Particularly small protein molecules, and in this case, T4 is an amino acid. It's based on tyrosine. So it's not a protein. It's a very, very small amino acid or small peptide. It's likely to get filtered in the kidney. So by attaching it to a larger carrier protein, you help preserve the action of this hormone. Well, what defines this cell as a receptor or as a target cell? That is the presence of a receptor. The receptor may be of two types, and the first example on the top of the page is the type of target cell that has a receptor which is located within the cell. That is, it is an intracellular receptor for that hormone. This is characteristic of all steroid hormones And the exception is T4, which is thyroxine, which is a small amino acid. It is a tyrosine derivative. Both T4 and all of the steroid or lipid hormones have the ability to diffuse across the membrane. They diffuse across all cell membranes. So what makes a target cell is the presence of this specific receptor. So only cells which have receptors for these compounds moving in will be responsive. The binding of this receptor to the steroid molecule, or in this case to T4, produces a complex which then migrates into the nucleus. Can you take me out of the picture for a second, Bo? No, there we go. So this is a diagrammatic representation of the nucleus. You see that the receptor is coming in, and it is this receptor hormone matrix that then produces the production of RNA that goes out to the endoplasmic reticulum and produces proteins. So you can see why this is a relatively slow process. That is, you must wait for this nucleus to generate the RNA, and the RNA then has to go out to the ribosomes. You have to produce a new protein. But it is that protein then that produces the response of this cell to that hormone. And you note here that this is on the terms of hours or days, to remind you that the production of these proteins is done on the rough endoplasmic reticulum. This is true of steroids and thyroid hormones. Okay, now you can bring me back. And we'll look at the other type of action, and that is characteristic of our peptide hormones, protein derivatives, and what are called the catecholamines. Well, you see the word amine on there, so you know it has something to do with proteins. And the catecholamines that we have already talked about include epinephrine and norepinephrine. Norepinephrine is a primary neurotransmitter. Epinephrine is primarily an endocrine. They are both examples of catecholamines. There is another one, dopamine. Dopamine is used pharmacologically. I mean, it's present in the body, and it has endocrine function, but it's also produced pharmacologically and used in the treatment of a number of nervous disorders. That, too, is under a group of compounds called catecholamines. So what distinguishes a lipid hormone action, lipid and thyroid hormone action, versus peptide and catecholamine? That has to do with where the receptor is located. Peptides or proteins have difficulty moving through membranes. Steroids, because they're lipid, move by simple diffusion. In order to identify this cell as a target cell, it must have its receptor located on the extracellular membrane. So steroid, intracellular receptor. Peptide and catecholamine, extracellular receptor. Now, hormone binds to this receptor, and in this example, it's the type of receptor that activates the G protein. Remember that from like way back when? This G protein is this on the intracellular side. It then moves to activate another protein called adenyl cyclase. Adenyl cyclase then takes and converts ATP to cyclic AMP, and it's cyclic AMP then that activates and turns on inactive proteins. Well, you don't have to produce the protein in this example. The protein already exists present in the cell. It merely needs to be turned on. This process is rapid, so we can see a response to peptide and catecholamines on the order of seconds or minutes as opposed to hours or days. You don't have to produce the protein. Remember that when we were talking about these cell kind of modulating these receptors and producing these second messengers, we talked about a cyclic AMP as being a second messenger that is something that is communicating what's happening on the outside of the cell. And the other example, if you could take me out of the picture for a minute, Bo. Let's turn docking off first. Okay. So here's the other example that we used in terms of modulating an extracellular signal to an intracellular change, and that is the presence of, again, a G protein, but altering calcium channels. And calcium then, based on the concentration gradient, moves in. There's this calmodulin molecule that binds, and it is this calmodulin calcium unit that then goes to turn on enzymes. In both these cases, the production of an active enzyme takes literally seconds or minutes. Okay, you can bring me back. Neurotransmitters, they would produce and induce a response. You needed to be able to turn it off. We talked about some of the enzymes that were involved in turning off neurotransmitters. Acetylcholinesterase, its function was very important in terms of terminating neural responses. Same thing with hormones. High levels of hormones for prolonged periods of time lose their signaling ability. We have what's called down regulation. So if hormones are present in large concentrations for prolonged periods of time, the receptors become less sensitive. A reduction in the number of receptors, a decrease in the affinity of the receptors for the hormone, meaning that the hormone's effects are attenuated or blunted. It simply loses its ability to signal changes in the body. So we need to get rid of these hormones. We don't want them circulating around all the time. The two primary ways is by metabolism in the liver, so there's liver degradation, and also excretion, so elimination of hormones in that fashion. Mechanisms of hormone regulation. So that's plate 115. Oh, again, we want to be able to turn things on and off. And I'm going to expose you to some of we talk about feedback, negative feedback, and positive feedback. So let's look first at a neural feedback regulation. And feedback just means sending information back to the cells from which the signal originated. So here's your endocrine system. Right? It could be a neural, it could be a neuroendocrine, it could be a neural system. But it's sending out information, so there's output. That output has to be sensed in some fashion. That is the body responding? Am I running fast enough? So there you are, cave bear's running at you, and you start running. Optically, you see the cave bear is gaining on you, so what you want to do is you're using that feedback information to modulate how the system responds. So let's give you an example. Let's talk about negative feedback. Negative feedback is where the system puts out a signal. So the output of this system is increased. It could be neural, could be a hormone. Think of it, how about heat? This system is not an endocrine system, it's not a neural system. This is the heater in your house. You turn up the heat in the morning. So the heat goes out, heat inside increases. Well, you have an elevation in house temperature, which changes the contraction of the little spring inside your thermostat. And what it tells the heater is shut off. So this input is what we call negative input. It's a negative feedback. The consequence is the heater shuts off. All right? So this is decreasing output. If it's negative for too long, the heat production is coming out, it's very, very low, and the output spring changes, comes back, and now it's positive feedback. So you have this negative and positive feedback control to regulate output. Now, if this is a hormone, think this is normal production. You've got the hormone coming out. Let's say the hormone's epinephrine. And you go, I need to increase my epinephrine output. Well, epinephrine output increases, output changes, heart rate goes up, fat mobilization goes up. All of those changes tend to bring the system back into normal. So negative feedback is an important regulatory mechanism. Positive feedback systems produce a disequilibrium. What that means is that all of the output goes to further stimulate output. So think about a heater. You turn the heater on, heat in the house goes up to 80, positive feedback goes back and turns on another heater. So the house goes up to 110. And you can see why this is sometimes called a vicious cycle. Levels of hormone regulation. The first example is the simplest. You have an endocrine gland or cell. It produces a hormone that gets dumped into the blood. It circulates to a target organ cell and produces an effect. In this negative feedback system, we oftentimes have more complex levels of hormone secretion. So, for example, in this one, the complex hormone involves a target endocrine gland. Can you take me out of the picture for a minute, Bo? There's the target cell, like that. But now what we've done is we've added another layer. And in this particular example, it is a pituitary gland cell. So this is a nerve cell, a modified nervous tissue cell, located in the brain, which produces a hormone which stimulates this cell, this hormone cell. Because this is a supportive hormone, right? So in this fashion, it is a supportive. It's what we call a tropic or trophic hormone, one that supports the function of this cell. This cell is producing a hormone. But in addition, maybe I should bring it up. So here's this hormone being produced by this endocrine gland. That's the target cell. This hormone not only produces an effect in this cell, but it has a negative feedback on this cell. That is a way of modulating or regulating the output. What it's telling this cell is, yes, I'm getting a response. We can further complicate it. Here's an endocrine gland. Here's a tissue which is producing a trophic hormone. And here's another tissue above it that is also producing a hormone. So there's this feed-down system. This hormone comes down and stimulates that gland. This gland produces a hormone that comes down to this gland, stimulates this gland to produce this hormone. Both can have negative feedback. And I think this is probably best exemplified where you have the hypothalamus in the brain. So that's kind of this cartoon. This is the hypothalamus, which has an impact on the pituitary, which then has impact on other endocrine glands. And we'll give you specific examples of that. So let's start with that. Let's start on plate 116. And look at the pituitary hypothalamus. I get it all on. So we're going to go in and do a sagittal section. We're going to cleave the brain, spread it apart. In lab, you should be looking at the pituitary gland and the hypothalamus. It's located at the base of the brain. The hypothalamus sits above the pituitary. The hypothalamus is neural tissue. The hypothalamus is connected to the pituitary by a stalk, what is called the hypophyseal stalk. So here's the hypothalamus, here's the stalk. This is all neuro tissue. The pituitary gland is also called the hypophysis. All right? To complicate matters. So this is the hypophyseal stalk, that is the stalk that connects the hypothalamus to the hypophysis or pituitary gland. The pituitary gland is divided into basically an anterior and a posterior lobe. Now in lab you'll recognize that there is in fact an intermediate lobe demonstrated here. So here's the anterior lobe, an intermediate, and then the posterior lobe. The anterior lobe is sometimes referred to as the adenohypophysis. The posterior lobe is referred to as the neurohypophysis. So there is the adenohypophysis and the neurohypophysis. Embryologically, the adenohypophysis, or anterior pituitary, evolves from the floor of the gut. There's kind of this involution and movement of gut tissue into this, so it's modified epithelial tissue. They join or fuse with a down-pocketing from the brain, which is neural tissue. So the posterior lobe produces, the posterior lobe is composed of modified neural tissue. The anterior, or adenohypophysis, is composed of modified epithelial cells. So there's two functions. The posterior pituitary is an extension of the hypothalamus, and that's represented in this diagram right here. So this is the hypothalamus. This is the posterior pituitary. And there is a connection from the hypothalamus to the posterior pituitary. This is a stalk, a hypophyseal stalk. And in this stalk runs axons, the cell bodies of which are located in the hypothalamus. So here's the soma or cell body of this neural tissue. It has an axonal projection down through the hypophyseal stalk, and it terminates at a capillary bed in the posterior pituitary. These are neural cells. They are producing a compound which is released from a neuron, goes into the blood. That is a neurohormone. Here's kind of the cell body of this neurosecretory cell. The hormone is actually produced up here in the cell body. It is then transported by axonal transport down. Can you remove me from the picture just for a second? Transported down the axon to the synaptic terminal. It is released, again, there's our friend calcium, just as we talked about the release of neurotransmitters for acetylcholine, for epinephrine. It's modulated through calcium influx. When this cell fires, sends an action potential down. This action potential reaches the terminal synapse, changes the permeability. Calcium comes in, and that calcium then, through the synapsin molecules, you remember any of this? I hope. There, that vesicle then fuses with the presynaptic membrane, dumps its compounds, but instead of going across to a receptor, this chemical compound goes into the blood and is transported into the circulation. Okay, you can bring me back now. A collection of cell bodies in the peripheral nervous system was called a ganglion. So we have this sympathetic ganglia, parasympathetic ganglia, because the collection of those cell bodies, that is the second or postsynaptic cell, the collection of cell bodies within the central nervous system is called a nucleus. So there's lots of nuclei, cell bodies that have a number of common functions. Sometimes they are described by their anatomical location. So if we look at where this is, and again, you can help appreciate this in lab. The hypothalamus is sitting right up here. The optic chiasm, and so there's the eyeballs right there. Optic nerve comes out, and it crosses at the optic chiasm. That crossing is right there, right underneath a portion of the hypothalamus. So in fact, there are a collection of cell bodies that are located above that chiasm. That is the supraoptic nucleus. So these cells right here, supraoptic nucleus. There is also a collection of cell bodies, distinct cell bodies, associated with the ventricle. These are the paraventricular nucleus, right? Paraventricular nucleus. Cell bodies in the supraoptic nucleus produce one hormone. Cells of the paraventricular nucleus produce another hormone. One hormone that you've heard a lot about in detail, and that is antidiuretic hormone. So when we talked about the kidney, we talked about a hormone called antidiuretic hormone. Now, in fact, it is produced in the hypothalamus, but it is secreted from the posterior pituitary. Stimulus for the release of antidiuretic hormone, fluid balance. So there's your input, a loss of fluid or an increase in electrolytes produces a sensory stimulus. Blood circulating through the hypothalamus is sensed. When concentrations of electrolytes increase, that then stimulates these cells to release its hormone. And the target tissue for the release, or for ADH, you recall, is the kidney, where it modifies the permeability of the collecting ducts and increases water reabsorption. The other hormone is oxytocin. So the paraventricular nuclei produce a hormone. Both antidiuretic hormone and oxytocin are examples of peptide hormones. So their action is by stimulating extracellular receptors. Oxytocin has as its target tissue the mammary glands and smooth muscle of the cervix. It is a hormone whose primary function is relegated to female function, specifically parturition or birth. The stimulus for the release of these is sensory as well, right? The optic nucleus was responding to a sensory change, a change in chemicals. The paraventricular nuclei respond to a neural input, specifically stimulation of the breasts, the mammary glands, and stimulation of the uterus. So at parturition, or near birth, you have the child budding into the cervix. Well, that stimulation, that mechanical stimulation, is perceived by the walls, by sensory nerves, that then come back and feed directly into the hypothalamus. In response, the paraventricular cells produce oxytocin. Oxytocin, which has as its target tissue that very same tissue, where it produces contraction. So you're setting up for birth, for delivery or parturition. The fetus is now moving into the birth canal. It's stimulating the cervix. The cervix says, oh, I have something in here. I need to get it out. And there is then a positive system that comes back, feeds in, and produces contraction of those smooth muscles. You also see that with stimulation of the mammary glands. Now you have the fetus has been delivered. You now have a neonate. The neonate needs to be fed. The neonate then suckles. So there is a sensory stimulation of the breasts or mammary glands, which in turn feeds back on the hypothalamic paraventricular cells. They release oxytocin, which has as its target tissue smooth muscle in the mammary gland. It's what we call milk letdown. So, and we'll talk about milk production and mammary glands in more detail in reproduction, but you have all of these fluid producing cells that are producing large quantities of milk. You want to be able to deliver that milk, and so oxytocin is your friend. And what I think I'll do is I'll stop there and we'll pick up lesson 20.